Imagine a world where damaged organs and tissues are not replaced by transplants but printed. Sounds like science fiction, right? Well, let's explore the realm of bioprinting, a revolutionary technology that's inching us closer to that futuristic vision. Picture this, a world where waiting lists for organ transplants are a thing of the past, where the fear of organ rejection is non-existent. Bioprinting, in essence, is the process of creating cell patterns using 3D printing technology, where layer upon layer of cells are deposited to produce tissue-like structures. It's like building a house, but instead of bricks and mortar, we're using live cells. This is not just a wild idea, it's a real, tangible technology that could redefine the medical field, offering potential solutions to some of our most pressing health challenges. But how did we arrive at this point where we can even consider such an idea? The answer lies in the history of bioprinting. The journey towards bioprinting started decades ago, with the development of 3D printing technology. In the late 20th century, an innovation in the field of manufacturing was taking shape, and it was called 3D printing. It was a shift from traditional methods, allowing creators to transform digital designs into tangible, three-dimensional objects. This technology opened up a new world of possibilities. It was initially used to create prototypes, architectural models, and even custom jewelry. But as the technology evolved, so did its applications. The idea behind 3D printing is simple yet powerful. It's a process called additive manufacturing, where an object is created layer by layer. This layering technique could be used with various materials, not just plastics or metals. This realization sparked curiosity and excitement in the scientific community. The question arose, if we can print inanimate objects, why not biological ones? This idea was not as far-fetched as it seemed. After all, living tissues and organs are also built layer by layer, cell by cell. In the early days of exploring this novel concept, scientists started with the basics. They began experimenting with biocompatible materials, testing if these could be used as ink in a 3D printer. They explored Bioink, a substance made of living cells that could be used to print tissue. These early experiments were both challenging and exhilarating. They tested the boundaries of what was possible, pushing the realms of science and technology. There were setbacks and triumphs, with each step bringing us closer to the birth of bioprinting. Bioprinting, the process of creating living tissues using 3D printing technology, was no longer a mere concept. It was becoming a reality. This was not just about printing objects anymore. It was about creating life cell by cell, layer by layer. As scientists continued to explore the potential of 3D printing, they began to consider the possibility of printing biological materials. This was the dawn of a new era in medicine, an era of endless possibilities. The birth of bioprinting had begun. The leap from printing inanimate objects to living tissues was not an easy one. It was a leap that required not only technological innovation, but also a fundamental rethinking of what it means to create life. In the early stages of bioprinting, scientists encountered numerous technical challenges. The precision required to arrange cells in a three-dimensional structure was unprecedented. It was like trying to build a house out of bricks, but the bricks were alive, each with its own needs and behaviors. Furthermore, these living bricks or cells had to be kept alive during the printing process, which required maintaining the right temperature, the right level of moisture, and the right chemical environment. Add to these technical difficulties the challenge of developing a suitable bioink, a material that could serve as a medium for the cells to live in while also being compatible with the printing process. The bioink needed to be capable of nourishing the cells, facilitating their growth, and allowing them to communicate with each other. And then there were the ethical concerns. Bioprinting raised questions about the sanctity of life, the potential for misuse, and the responsibility of scientists in creating new forms of life. These were weighty issues, the kind that could not be resolved in a lab or a boardroom, but required public discussion and societal consensus. Yet, despite these challenges, the prospect of creating tissues and organs that could save lives and alleviate suffering was too compelling to ignore. The promise of bioprinting drove relentless scientific pursuit, pushing boundaries and challenging conventions. This pursuit was not without its failures. There were many, but with each failure came new knowledge, new insights, and renewed determination. And then finally, there were successes. The first successful bioprinting experiments marked a turning point, a moment when theory became practice, and the impossible became possible. They were the result of years of hard work, dedication, and an unwavering belief in the potential of bioprinting. Despite these challenges, relentless scientific pursuit led to the first successful bioprinting experiments. In the early 2000s, the world witnessed the printing of the first living tissues. It was a moment that felt straight out of a science fiction novel. But this was no fiction. This was the reality of bioprinting. The first bioprinted tissues were simple, thin layers, but they marked the dawn of a new era in the medical field. These first experiments in bioprinting were met with a mixture of excitement and skepticism from the scientific community. On one hand, scientists were thrilled by the promise of this new technology. The ability to create living tissues from a printer was revolutionary, presenting a plethora of possibilities for research and treatment. On the other hand, there were those who questioned the feasibility of bioprinting, citing concerns about the longevity and functionality of the printed tissues. The public reaction to the first bioprinted tissues was equally mixed. Many were fascinated by the concept of printing body parts, while others were apprehensive, unsure of the ethical implications of creating life in a lab. However, as the technology advanced and the potential benefits became clearer, public opinion gradually shifted towards acceptance and anticipation. So what did the creation of the first bioprinted tissues mean for the future of medicine? In one word, potential. The potential to create custom-made organs for transplants, eliminating the need for donor organs and the associated risk of organ rejection. The potential to study diseases in a new way, by creating diseased tissues in a lab and testing treatments on them. The potential to reduce the need for animal testing, by testing new drugs on bioprinted human tissues instead. The first bioprinted tissues were a stepping stone, a glimpse into a future where bioprinting could become a standard part of medical practice. They were a testament to human ingenuity and a symbol of hope for those in need of organ transplants or suffering from incurable diseases. With this breakthrough, the potential applications of bioprinting began to take shape. As we continue to explore this exciting frontier, who knows what possibilities await us in the world of bioprinting? Today, bioprinting is a rapidly evolving field with a wide range of applications. This innovative technology has opened doors to possibilities that were once the stuff of science fiction. But what exactly are these applications? And what barriers do we still need to overcome? Let's delve in. One of the most promising applications of bioprinting is in the realm of tissue engineering. Scientists have already succeeded in bioprinting simple tissues such as skin, cartilage, and blood vessels. This could revolutionize how we treat injuries and diseases. For instance, victims of severe burns could receive bioprinted skin grafts, custom-made to perfectly fit their injuries. This is not just a theoretical possibility, it's already happening. Beyond tissue engineering, bioprinting has significant potential in the field of pharmaceutical testing. Today, new drugs are typically tested on animals before human trials can begin. Not only is this ethically questionable, but it also doesn't always provide accurate results, given the biological differences between humans and animals. Bioprinting could offer a solution. By creating miniature bioprinted versions of human organs, often referred to as organoids, researchers can test new drugs with greater accuracy and less ethical concern. But it's not all smooth sailing. While the possibilities of bioprinting are vast, there are still significant hurdles to overcome. For one, bioprinting complex organs like the heart or liver is still a distant goal. These organs are incredibly intricate, made up of multiple types of cells arranged in precise ways. Bioprinting them requires a level of precision and complexity that we're still working towards. Moreover, there's the challenge of vascularization, the process of developing blood vessels within the tissue. Without a network of blood vessels, bioprinted tissues can't receive the nutrients they need to survive and function. Scientists are making progress in this area, but it's a major obstacle that needs to be overcome. Another limitation lies in the regulatory landscape. Before bioprinted tissues can be used in clinical settings, they need to be rigorously tested and approved by regulatory bodies. This can be a long and complex process, often taking years or even decades. Finally, there's the issue of cost. Bioprinting is an expensive technology, and bioprinted tissues and organs are likely to be costly. Ensuring equitable access to these potentially life-saving treatments will be a major challenge. Despite these limitations, the progress made in bioprinting is undeniably exciting. Every day, researchers are pushing the boundaries of what's possible, inching us closer to a future where printed organs are no longer a dream, but a reality. As we continue to push the boundaries of bioprinting, we move closer to a future where printed organs are a reality. What does the future hold for bioprinting? Only time will tell. But let's take a speculative leap into the realm of the possible, the exciting, and the transformative. Imagine a world where organ transplants are not limited by the availability of donors or compatibility issues. A world where a patient's own cells are used to create a perfectly matched organ, printed layer by intricate layer. This is not some far-flung science fiction fantasy, but a real possibility on the horizon of bioprinting. Researchers are tirelessly working towards this goal, and while we are not there yet, strides are being made every day. Similarly, think about the potential impact on personalized medicine. Doctors could prescribe treatments tailored to an individual's unique genetic makeup, eliminating the trial-and-error approach that can often accompany current medical practices. With bioprinting, it might be possible to test a patient's reaction to a drug on bioprinted tissue before administering it, thereby increasing the efficacy and reducing potential side effects. The future of bioprinting also holds promise in the field of prosthetics. Imagine bioprinted skin grafts for burn victims, or bioprinted bones to replace those damaged by injury or disease. The possibilities are as vast as they are exciting. Of course, the road to these advancements is paved with ongoing research and development. Scientists are constantly pushing the boundaries of what's possible, exploring new techniques, materials, and applications. They're tackling the challenges head-on, whether it's finding ways to vascularize printed organs or refining the precision of the bioprinting process itself. And let's not forget about the ethical and regulatory considerations. As with any groundbreaking technology, there are important questions to be asked and answered about how to use it responsibly and effectively. While the future of bioprinting is still being written, one thing is clear: it has the potential to revolutionize medicine. It's a brave new world we're stepping into, and bioprinting is leading the way. Bioprinting, a concept once considered science fiction, is now a scientific reality. This journey, which began with a mere idea, has transformed into a tangible tool that's beginning to reshape the medical field. So let's take a moment to reflect on what we've learned. In the early stages, bioprinting was but a concept, a notion formed in the minds of pioneers who dared to dream beyond the realms of traditional medicine. It was a vision born from the merging of biology and technology, a testament to the innovative spirit that drives scientific exploration. From this theoretical foundation, the concept of bioprinting evolved into a practical application. The birth of bioprinting emerged from the convergence of bioengineering and 3D printing technology, creating an entirely new field of scientific endeavor. It was no longer a question of if bioprinting could become a reality, but when, and the when wasn't too far off. The first bioprinted tissues marked a significant milestone in the journey. It was a moment of triumph and validation, as the first tangible products of bioprinting were brought into existence. From simple tissues to complex organ structures, the potential applications of bioprinting began to unfold before our very eyes. However, like any new technology, bioprinting is not without its limitations. The current applications of bioprinting are still in their infancy, with many challenges yet to be overcome. Issues around biocompatibility, structural integrity, and ethical considerations continue to be hurdles that need leaping. Yet these challenges are not insurmountable. They are merely stepping stones on the path to progress. As we look to the future, the potential impact of bioprinting is nothing short of revolutionary. Imagine a world where organ transplants are not limited by donor availability, where personalized medicine is not just a concept, but a standard practice. Bioprinting could well be the key that unlocks this future, bridging the gap between what is and what could be. To sum it all up, bioprinting is a journey. It's a journey that has taken us from the realms of science fiction to the cusp of a new era in medicine. It's a journey marked by innovation, challenges, and immense potential. It's a journey that's far from over. As we continue to explore the possibilities of bioprinting, we may be on the brink of a new era in medicine. The future is an open book, and bioprinting is poised to write some of the most exciting chapters. So, as we turn the page, let's look forward to what the next chapter of this incredible journey holds.